Welcome to Rubin On Point, a podcast about cancer, resilience, and being human. I'm Dr. Dan Rubin, and together we'll uncover the true power of resilience, the strength to recover, adapt, and thrive through life's toughest challenges. Each episode will bring bold ideas, transformative practices, and inspiring stories that redefine what health, healing, and what the human spirit can achieve. And you're going to meet some really cool people along the way. If you're ready to turn obstacles into opportunities and live with greater purpose and courage, you're in the right place. Let's get started. Your journey to resilience begins now.
Hi, Dr. Brandon Guida. I just want to say thank you to Dan here for bringing me in, and this is going to be a fantastic talk. All of the opinions and the things that I discuss are strictly personal. They're not representative of any of my previous, my current, or my future employers. Just want to get that out of here. Thanks.
Get ready for the heat because we are bringing another episode of Rubin On Point. I'm Dr. Dan Rubin, and I'm joined today by Dr. Brandon Guida, who has become a good friend.
Yes.
Through clinical medicine.
Absolutely.
And we're going to talk a lot about cancer diagnostics, specifically molecular diagnostics, because you indeed are a subject matter expert in cancer molecular diagnostics.
Indeed, I am.
So,
yeah.
What does that actually mean?
Oh, that's a good question. So, I mean, my academic pedigree is all over the place, but I've been in genomics and genetics for about 20 years now.
What's the difference between genetics and genomics?
That's another great question. So genetics would be the study of the genes of an individual organism, whereas genomics would be the study of everything in the DNA, not just the gene segments, but the dark DNA plus the RNA. So all of that would be genomics.
Okay, so you said that genetics is a study of just the cancer genes, but genomics is a study of other things associated, not just the genes, but the RNA, which comes from the DNA, and then the dark DNA.
Yeah, it's kind of genomics is kind of the computational analysis of the genome and everything that's happening and all the interactions that take place.
Okay.
And, genomic analysis, I think, there's still a lot of genetic analysis out there. You can do straight up genetic analysis, but I think most of the field has migrated towards genomics.
Is it fair to say that genetics just looks at what DNA, but not necessarily what's happening in genomics is what's going on in the body related to those genes?
Correct. It's the dynamics that's associated with the genome.
And that's important because we're in an era of precision medicine or personalized medicine. Absolutely. And a lot of it is based around interpreting the expression of cancer-related genes for the most part, I would say, because we do testing.
Yes.
Right?
Absolutely.
So before we jump in and talk about this testing, that many of our listeners be like, okay, I got that testing. I never really understood exactly what it was. So they're going to be happy to be listening to this. And so am I, of course. What's your background? How'd you get here?
My background is in infectious disease and microbiology, actually. So I'm a trained microbiologist, but in that regard, a lot of molecular biology, right? So that is translational for a lot of things. And then at one point, what does that mean,
translation?
So I think the skills are transferable.
Okay.
So, you do a lot of molecular analysis in microbiology, and I think those techniques and the paradigms and the concepts are applicable outside of micro. Okay. And I think I've kind of taken that as, my career is predicated on that. Yeah. So I'm now in the molecular biology world. I was in molecular neurobiology and neurogenomics for a while. And I think a lot of the research that I did there had oncology overlap, and that allowed me to transition.
What did you do in the world of molecular neurogenomics?
So I worked for Barrow Neurological and Phoenix Children's Hospital, and I worked there with Dr. Michael Kruer. He's the director of the Pediatric Movement Disorders Clinic for PCH, and we were studying the genetic etiologies of cerebral palsy and Tourette's and epilepsy. So we were doing deep dives into patients and their families genomically and looking at what causes, what could be potentially causal to CP.
Did you see some associations or causalities or both?
Mostly associations, but there is some causality. So there are some twin studies, but there was also some consanguineous familial analysis that we could do, and we saw that it was one specific gene that was passed down in this family, and everybody who had this particular allele had cerebral palsy or a variant of cerebral palsy.
Not necessarily a gene, but a part of a gene?
It was a specific mutation in a specific gene.
Okay.
Yes, that was inheritable.
And that one led to cerebral palsy,
Correct.
But did not lead to anything else per se?
So there is some haplotype interactions, right? So there are other alleles that a person could have that could influence the outcomes. But yes, they were all CP-like.
So then the big question is, can you go in there and fix that?
Yeah, that's a really good question. And I think, the technology, I think, is there now. We have some fantastic abilities to do in vivo gene editing, right? We're seeing a lot of that types of
While
technology
there's
come
a
out.
fetus in the mother's
Yes,
womb?
that is a possibility.
Okay.
And so, for example, I think relatively recently there was some work that was published, and the media sensationalizes everything, but we now have the ability to silence the extra chromosome 21 in trisomy 21. So
Which is Down syndrome for some people.
Correct, and Down syndrome. So there is the potential to actually prevent Down syndrome in utero.
Wow.
Which is a super.
Has it been
interesting.
tried yet?
I don't know off the top of my head if someone's actually done it. I know that we can do it in vitro.
Got it. So then is there a full circle to this? Meaning, is there a way to look at, say, a BRCA gene or like an hereditary cancer syndrome and maybe do some gene editing? Is that ethical? Is that something that you're able to talk about?
Yeah, I mean, the ethicality in the case of BRCA mutations, in my mind, I would say that there would be much less ethical debate in that regard, as opposed to, for example, in the Downs argument. There are a lot of groups that, there are systems for individuals that have Down syndrome, and they're advocating, saying, hey, these people have great quality of life. Why would we eliminate that? What's the problem with Down syndrome? Why are we looking at this as something that needs to be cured, right? I think that we don't have that level of advocation in the BRCA community because it is such a pathogenic disorder. Right?
Meaning that there would be less inclination to edit in utero a BRCA gene, or you're saying there's more inclination?
More inclination.
Okay, that's what I was thinking.
More inclination, yes.
Got it.
Yeah.
Got it. So then fast forward, and what did you do before your current position?
So I entered into the medical affairs arena. So all major biotechnology and pharmaceutical companies, they have a medical affairs department, right? And this department specializes in the communication, the objective scientific communication of company science and supporting science to clinicians, right? And that's the sole kind of job of medical affairs, to go out into the field, talk to providers, gain insights into the drug, if it's a drug company, or into the diagnostic test, if it's a test. Bring those things back, and then help improve that test, right?
Okay.
So that's medical affairs. So I've been doing that since COVID. So that was kind of the transition.
So 2020, 2021.
2020.
Okay.
Yeah. So I've been in medical affairs. I've worked in regenerative medicine and orthopedics in medical affairs, and then prior to my current role in oncology diagnostics, I worked for a CRO. So that's a contract research organization specializing in genetic analysis for clinical trials. And we were doing a lot of cell-free DNA analysis. Again, so this is kind of the technology and the modality when you're doing oncology diagnostics using the blood. We're looking at cell-free DNA.
Okay.
Right.
So is it fair to say that right now your current position is in the world of liquid biopsies?
Yes, absolutely. Yes.
Liquid biopsies to me as a clinician means that we're using a liquid. We're using the blood as the sample agent, and we're sending it to the lab for analysis to look at the genomics of the landscape of what's going on related to the cancer.
Correct.
That's a liquid biopsy. And they use the word biopsy because is it diagnosable? Because a biopsy usually, what is a biopsy? Biopsy is taking a sample to be able to diagnose a condition.
Sure.
Yeah.
Any biological sample, right?
Yeah.
You could take a biopsy of any fluid or any tissue technically, and then do a molecular analysis of that. And depending on the level of evidence that's out there that's been generated, right, that can dictate whether or not something is diagnostic.
Are we moving towards an era where excisional or fine needle aspiration or sort of aggressive biopsies could be maybe not eliminated completely, but minimalized, and we could move towards liquid biopsy, just a simple blood test to diagnose people with cancer?
Yeah, absolutely. I think we're right in the midst of that paradigm switch. I think there was something that I saw on my LinkedIn feed where there was a paper that was put out comparing tissue NGS, so tissue next generation sequencing, which is the gold standard still, and it's been the gold standard for 40 years, right? Looking at the DNA and the nucleic acids in a tumor biopsy, and then saying,
"Oh, go ahead." Sorry.
Right? No worries. And then saying that we found this mutation in this person's tissue.
So NGS has been around for 40 years.
So PCR has been around since 1987 and 89, right? And then the early 90s, we discovered some of the genes and we were piecing on those out. So next generation sequencing came a little bit later, post Human Genome Project in the early 2000s. So we've been doing NGS, so straight up NGS on tissue for about 25, 30 years now.
Got it. Yeah. So they're still testing using PCR, right? In fact, some of the COVID tests were based on PCR technology. Yep. There are still some liquid biopsies that are in play. Some of the companies still use PCR. I know some of the methylated DNA capturing to look at trying to capture cancer in the first place. There's some screening tests. We had Dr. Richard Bernert from Episeq here talking about he uses a PCR test for early detection of cancer. But you're more an expert in NGS or next-gen sequencing.
I mean, my first foray into molecular biology was doing PCR. So, I mean, I've been doing PCRs way longer than I've been doing NGS. And I think, I think anybody's in that same boat. If they're an NGS expert, they probably started with PCR. That's kind of that.
Is there one you
material.
prefer for cancer molecular diagnostics?
So I think PCR isn't going anywhere. It's a foundational molecular technique, and I think it's very specific and it's very sensitive for a very specific piece of DNA.
So you can't just go looking for stuff. You have to know what you're looking for.
It's not fishing necessarily. You need to have bait, and your bait is your primers, right? In PCR. So you need to know exactly what you want to find. But if you have that data in mind, it's very sensitive.
Got it.
Yeah.
Is one less expensive than the other usually?
PCR is dirt cheap. Comparatively speaking,
What does PCR stand for?
Polymerase chain reaction.
And so what does that mean?
So it means that DNA is a polymer, right? It's a chain of A's, T's, C's, and G's that have all been linked together, right? And so we can take a piece of DNA and then we can add primers, which are other pieces of DNA, which are complementary. In other words, they will bind to a specific sequence, right? And then what happens is we add some enzymes and we add the A's, T's, C's, and G's, and basically we do this reaction where we can fill in the gap. Right, based on a piece of DNA. But
it's real, even though we're filling in the gap, but it's not artificial. Is it artificial in that we're creating it, but it's still legitimate?
Yeah, it's like taking a piece of a page out of a book and photocopying it. I mean, it's the same thing.
It's the same thing.
Yeah.
Okay.
Yeah.
Got it. Yeah. And it's that specific. I mean,
specific.
Yeah. So for the audience, so NGS, you said, stands for next gen sequencing. And then what does it actually mean? Why is it next gen? And what do you mean sequencing?
Yeah, that's a good question. So first gen sequencing, I think, would be most likely considered by most molecular biologists to be Sanger sequencing. So it's called Sanger sequencing based on the scientist, I think, Fred Sanger, who developed this. Yes. And so this was also called dideoxy chain termination sequencing, long name, right? And so it's much easier to say Sanger sequencing. And that's basically a modified PCR, which the A's, T's, C's, and G's in that reaction, they have fluorescent tags on them. So when an A happens, and they're specially modified enzymatically, so when the A gets incorporated in the PCR reaction, it stops, it chain terminates. And so what happens is this specialized PCR reaction creates fragments of every single size because it's random where the chain terminates. And then we can run that on a specialized sequencer, scan it with a laser, and look at fluorescence, and then we can get the actual A, T, C, or G sequence in that lane, right? So that was first generation sequencing. Now the issue with that is that you could really only get between 600 and 800 bases of information per run, right? So it was a very arduous process to set up and relatively expensive compared to just regular PCR to run, and you didn't get very much data from that. So people, and when I say people, a lot of the people who started Illumina, for example, one of the biggest NGS companies in the world, right? And those guys all got together, and many others in the world, and they say, well, how can we do this faster? How can we generate more information less, and at a much higher quality? And they came up with next generation sequencing.
And they built the Illumina sequencer.
There were predecessors to that as well. Some other types of sequencing modality like Celexa and things like that. But
what do you work on currently?
So currently, the vast majority of clinical assays run on Illumina instruments.
Okay.
The newest kind of Mercedes-Benz of sequencers is the NovaSeq X Plus.
Okay.
And it can run an entire human genome in less than a day.
How many bases can you get with that on a single run? You said 6 to 800.
25 billion, I think, is one of their chips on a single run. So it's a lot.
We are
of data.
in the next gen.
We are in the next generation,
That's
right?
great.
And just for context, the human genome is 3.8 billion letters, right? And the Human Genome Project, which was predominantly done doing shotgun sequencing via the Sanger method, right? Started in 1998, was published in 2001 at a cost of over $2.5 billion.
Wow.
Right?
And now we're just, what, $1,000 bucks?
$1,500
Yeah.
for a single human genome in one day, less than a day.
Boom.
It's crazy.
Line up, get
Yep.
sequenced,
Yep,
and you're
that's
good
right.
to go.
Exactly.
So then before we continue on, what is dark DNA?
That's a good question. So 99% of the human genome does not code for a protein. So
99%. Let's back
99%
up on
about,
that.
right? 99%.
What is it? Does it encode for anything?
It does. So back in the day, do you remember your school days, right? When you took genetics or something, then they said, well, most of your DNA doesn't code for anything. It's only your genes that code for something important, right? And back in the day, they would call it junk DNA, right? We know that's horse crap, right?
Well, it's there for a reason.
Right, it's there for a reason, right? Biology is parsimonious, and there isn't going to be a bunch of stuff there if it's not being used for a reason.
Right. I mean, absence of evidence doesn't necessarily equate to absence of meaning.
100%, right? So I was always quite dubious of that junk DNA in school, and I'm like, this doesn't make any sense, right? But now I think we know that.
Did you get kicked out of class?
No, I didn't.
Okay.
I kept my mouth shut when I was supposed to.
Right. And now look.
And now I just talk about everything, right? So I think this dark DNA is going to end up bearing a lot of fruit clinically. That's my personal hypothesis. I think that there's a lot of information that can be gleaned from that. I mean,
Are we analyzing it now?
we are. We're analyzing a lot of it.
Are you reporting it?
No.
Okay, that's what I thought.
Yeah.
Getting ready, figuring out what it means.
Yeah.
The data's there. You'll be able to look back, do a retrospective analysis when it's ready.
I think a lot of people who are specializing in the dark genome, as a lot of people call it, they're trying to functionally categorize all of that sequence. When is it transcribed? What is it doing? If I mutate it, what happens in the cell? So they're doing, a lot of functional experiments. And I think it's going to take a number of years before those bear clinical fruit. But I think it's going to be there. I think it's important, obviously.
It has to do something with something.
100%.
So what did you mean that not all your genes code for, what was it? You said not all your genes code for DNA? Or
Not all of your DNA has genes.
Not all of your DNA has genes.
Correct,
Some of
correct.
your DNA doesn't have genes.
Most of your genes is not the canonical gene, right?
Okay.
When you're in school, again, you're taught that the one gene, one polypeptide hypothesis, right? It's one gene makes one protein. That protein goes on to do whatever phenotypically it's supposed to do in the cell, right? But we know that humans only have between 20 and 25,000 actual genes. That number goes up and down depending on how you define a gene. But I think that the rest of the DNA, the other 99%, right, is mostly regulatory and structural. And that's what.
So it codes. It's the DNA is there, and it's a structure support, like a scaffolding for the actual genes.
Could be scaffolding. It could control how the chromosome lines up in the nucleus, the three-dimensional chromatin structure within the nucleus. Could control what connects there, for example, to read the DNA.
So it's still structurally DNA, but it doesn't encode for genes, and therefore encode for proteins.
It's not transcribed, so it's not turned into RNA,
Okay.
right? But it's doing something fundamental to the way DNA functions.
You can't just take it away
Right.
and think we're going to function normally.
Right.
Interesting. I mean, we wouldn't be able to. So when we wouldn't be able to replicate. So when we replicate our DNA, that's not just replicating genes; it's replicating genes and all the other stuff.
Correct.
I think it's funny to be like, oh, it's just junk DNA. We don't know what it is. Like, what?
Yeah, what?
I never thought about it back then, and and certainly not until now. But thinking about it, it's like kind of laughable to really think that would be what it would be taught instead of something like. And maybe this is an issue with schools in general, and I'm not shy to speak out about that.
Sure.
But just be like, class, we don't know what this is. We're humans, and it's part of us, and therefore it's important. And science is looking to figure it out. Because what does that say to the child? Just be like, oh, there's things there, but we don't have to. You can just ignore them.
Right.
So
right.
anyhow.
Yeah. And,
that's not a tangent.
Right. Yeah, that's not a tangent. It's all related, right? It's all related. It's DNA. It's related. Yeah, 100%.
So then fast forward. Now you work in this field and you're an expert in helping people analyze tests. You help our clinic. You help me a huge amount. And we have this thing called cell-free DNA and circulating tumor DNA. And are those the same things?
They are not. We have known about cell-free DNA for, I believe, about 40 years was about the first publication, right? When we realized that, hey, if we spin down blood and we look at the plasma and we look at what's in the plasma, there are these fragments of DNA that are just floating there.
Some of them could be dark DNA?
Could be. Okay. Could be random representations of all of the locations of DNA, right? So as our understanding of cell-free DNA has evolved, right, we now know that all of our cells, not all of them, but most of our cells have a turnover rate, right? They are destroyed, they die, they're replaced, they go through this
housekeeping.
Housekeeping, replicative cycle. And when those cells die, they break open and they release stuff from inside, right? And we know that, for example, apoptosis, right, is programmed cell death. So when the cell is like, I'm really damaged, I can't be repaired, I'm going to die, right? It commits cellular suicide.
That's a program written in there. It's programmed in.
Correct.
And sometimes that goes bad.
Sometimes that goes
we'll
bad,
talk about
right?
that too.
Yeah, that's a hallmark of cancer, right? Okay. But there's also other modes of death. But in most of these modes of death, there's a very formal, stepwise thing that happens to DNA, and it gets fragmented by innate internal endonucleases and exonucleases in the nucleus.
What are
and it
those?
Gets fragmented. Those are so an endonuclease is an enzyme that cleaves DNA from within the middle of the sequence, and an exo means
breaks
it has
it
to
down.
Be correct.
Right.
Like cleaves it, cuts it like a pair of scissors.
Okay.
Right.
Got it.
So it can cut the scissors. It's a molecular scissors. It cuts the DNA right in the middle, right, into two strands. And exonuclease is one that can only cut from the end.
Oh.
So it can trim from the edge of the DNA, and endo can cleave from inside. Okay. Endo versus exo.
And these live where?
They live everywhere.
Okay.
They're all over the place.
Are they in the blood?
They are in the blood. They're in the cytoplasm. They're nucleus. They're in bacteria, so they're all over your desk.
Okay.
There's nucleases everywhere.
Everywhere.
Right?
Okay.
Everywhere.
So just in case a piece of DNA falls out, that's going to get cut up.
Yep.
Okay.
And DNA is rather robust, but RNA, on the other hand, if you talk to RNA in the wrong way, it goes and it breaks down. So RNA is very, very
doesn't
fickle.
require nucleases.
Right. It does, but those RNases are everywhere.
Okay.
And so
they're more ubiquitous?
Yes.
So like everywhere. Okay.
Yeah. So if you were to take, and you may have done this experiment in school, but you take strawberries and you take the DNA out, right? And you do any DNA extract. If you were to drop that DNA on the floor, right, mix it up with some dirt and gravel and pick it back up, you could still extract DNA and get great sequence data from that. But if you do that with RNA and drop it on the floor, the nucleases would chew it up.
So fragile.
So fragile.
Got it.
So fragile.
So that, there are like exome sequencing and there are RNA studies, and I know a lot of the testing houses are doing this, but they're holding the data. It's not necessarily ready for prime time, but it sounds like it's much harder to extract and interpret just because of the fragility of RNA. And and when you're looking at RNA, is exome sequencing looking at RNA?
So genes are broken down. A gene is broken down into two basic types for the most part of sequence. There's introns and exons.
Okay.
So the exons are the part of the gene that actually code for amino acids and protein.
Okay.
The introns are internal sequences, and they're used for other things like regulatory and alternative splicing. An exome, a whole exome sequence, is sequencing only the exome portions of genes.
Okay.
It's not a whole genome sequence.
Got it.
So you've got a whole genome sequence which sequence everything, including the dark
Right.
DNA. So you get 99.9% of all the A's, T's, C's, and G's by whole genome sequencing.
Okay.
Right? Whole exome sequencing is one level down where you only get the exomes of all the genes.
But that's what's put out into the world of the body by the cells, correct?
Yes, that is what is perceived by most as being the most important portions of the DNA because it's coding for the proteins and the proteins are doing the work.
And that's so that's what's supposed to be happening in the body.
Correct.
But now we're saying there's actually other layers that we're now having understanding of and how that plays a role.
Correct.
So before we actually understood that, maybe we just were like, okay, the exomes are the most important part.
Right.
So we're getting away from that then.
Right. So exomes are super important for many aspects of genomics and in the clinic. So for example, in my work in the neurogenomics department at Phoenix Children's, we needed exomes because we needed to look at what variants, what specific alleles were inherited from the parents. We had to look at all of the exomes. But in the case of cancer, we know that for most indications, there is a subset of very specific mutations and only a handful of genes. Okay. So it's quicker, logistically, it's cheaper financially to do a panel-based NGS assay to only sequence the genes which are most important in cancer.
And so there are some companies that will sort of have a less breadth but more depth.
Correct, absolutely, and that's a depth is a term that's used in the NGS context, and the depth of sequencing refers to how many times we have sequenced a region of DNA, right, in an individual read. Right? So
why is that important?
The more fragments we sequence at a locus, the more confident we can be in what the bases at that particular DNA locus actually are.
Got it. And breadth refers to what?
Breadth would be the number of reads across a region that we actually have.
Yeah. So it's expensive to have a bigger breadth, and it's certainly more expensive to have a greater depth. So sometimes ordering a really focused panel but having a real of depth means it's going to have really great sensitivity and specificity, but you might not capture certain things that you weren't necessarily looking for.
Correct.
Per se.
Correct.
So,
and your whole exome is going to be your high breadth, lower depth.
Got it.
Generally.
So then you could actually, once you find something on the breadth, you could say, okay, we're going to take it to a deeper level.
Correct.
So cell-free DNA and circulating tumor DNA, you said, were a little bit different.
Yes. We got off our tangent. Now we're circling back, right? So cell-free DNA we've known about for a long time, and it's used clinically for a lot. In the oncology setting, we know that, and again, we're going back to this idea that our cells are all turning over and dying, right? Cancer cells do the same. So do we have the technology to go into all of those DNA fragments that are in your blood and just pull out fragments that contain oncodriver mutations, for example, right? So EGFR is a gene that's very commonly mutated in lung cancer. Can we find specific mutations in EGFR in this cell-free DNA and attribute that specifically to tumor? And it turns out that we do have the sensitivity to be able to do that. So we can actually go into a person's cell-free DNA and we can get a fairly quantitative gauge as to how much cell-tumor DNA derived is in there.
Okay, so again, we talked about cell-free DNA and CT or circulating cancer tumor DNA.
Correct.
And the difference is exactly what?
So ctDNA is going to be specifically attributed to and derived from tumor cells.
Yes.
So
tumor cells in the blood or tumor cells in the body that and the DNA gets put into
That's
the
a good
blood?
question, and I think it could be both. Okay. So there is ctDNA in the case of hematopoietic cancers, but there's also ctDNA in the cellular tumor sense as well.
Okay.
And if a person has two indications, it would be very difficult to tease.
Well,
that
somebody
apart.
has like chronic lymphocytic leukemia with nodal activity,
Correct.
which means that they could have solid and hematologic
absolutely.
activity. We're not really looking at CT and cell-free DNA in those cases, at least
not yet.
Not yet.
Yes.
But let's say somebody with a non-small cell lung cancer, because you brought up EGFR. Sure. Would I look at the CF or the ctDNA, or would I look at both independently?
Generally, we look at them independently, but the cfDNA concentration naturally fluxes. Your liver clears that. The half-life of cell-free DNA is very short. And so it's
minutes?
Minutes to hours, depending on the fragments. Yep.
Got it.
So generally, when you're looking at CF or ctDNA, it's a fairly accurate real-time snapshot of what's happening in a patient right then when you take the sample. But yeah, so I think
Can I interrupt you?
Sure.
So if somebody had just had a chemotherapeutic agent given, 6 hours before, let's say that let's say it was a platinum, it had maybe a 30-minute half-life. At a cfDNA test at 6 hours post chemotherapy might look different than 6 days post.
Absolutely.
Okay.
There's a lot of data that shows that, and there is treatment-specific kinetics that are associated with different treatment modalities. So chemotherapy, radiation, for example, when you're destroying a bunch of cells and they're flooding their plasma with their DNA, right? When you
so
blow
it's
it up.
important too for patients to be like, oh my God, my cfDNA percentage went up. And be like, okay, well, when did you get this?
Right.
You know,
Right.
So that would be important for people to know and to make sure that they're getting it drawn at a close to optimal time.
Yeah, and in the targeted therapy and the chemotherapy, in the solid tumor sense, generally we recommend, and the most of the data suggests, 4 to 8 weeks post cessation of your last treatment.
Okay.
That's
what if somebody's on a weekly chemotherapy treatment, yet their scan shows progression? Would that be an okay time to draw?
I think that there is data suggesting, and it depends on what you're looking for. If you're looking for the presence of specific mutations, I would say yes. But if you're looking to utilize ctDNA or tumor fraction, like is the quantity of ctDNA there, that's what we call the tumor fraction. If you're looking to use tumor fraction to actually monitor what's happening,
yes, let's talk about that.
That might be a tricky scenario.
Okay.
Right.
So can you back up before you explain the mechanism? You said you used the term tumor fraction. Can you please explain that, how it's clinically, and then let's talk about how we would use it.
Yeah. So tumor fraction is generally defined as the percentage of ctDNA a patient has out of their cell-free DNA.
Okay.
Right. So if a tumor fraction comes back at 20%, that means of all the cell-free DNA that we got from the patient's sample, 20% of it is tumor-derived.
Got it.
Right?
And
That's
the other
pretty
80% is
normal.
Got it.
Right.
Okay.
Normal. Yeah.
So then if that percent goes up, well, let me back up. Let's say somebody was doing a water fast and they were having maybe some cell turnover or, cell senescence or some autophagy, or they were undergoing some, biohacking, and then they stopped. You could see some difference maybe in some overall ctDNA, and that might skew the cell-free DNA percentage. If you could see an increase in your percentage of ctDNA as compared to as a percentage of your total circulating
or vice
cell-free
versa.
DNA, or vice versa, depending on. So there are a lot of things now, especially because these things are becoming popularized, and there are different types of cell turnover even in the normal world. So we have to take these into account.
Yeah. And I like to think about this, and this is the way I will explain it to clinicians sometimes, is think about ctDNA generally and hopefully is a very small percentage. Right? Tumor fraction should be very low.
Like,
hopefully.
Would you say 0.3%?
Could be 0.3%, could be 0.01%.
Okay.
So
but it's significant at that level.
It can be.
Okay.
Yeah. So if you're looking at this tumor fraction percentage, and usually it's pretty low, and you think about the needle in the haystack analogy, right? So you think about the cell-free DNA is the haystack, and you're looking for ctDNA, which is a needle, right? After radioablation or after chemotherapy, you have now just increased the size of the haystack, because all that normal DNA is now being flooded into the blood. It makes it much more difficult to detect the tumor-derived DNA because it's so small.
So you work in the world of using ctDNA or tumor fraction as not a pregnant, but to follow. I mean, there are tests out there that are looking at this ctDNA,
Absolutely.
like for MRD, looking for minimal residual disease when a patient is not on conventional treatment. I mean, maybe in the case of somebody with breast cancer, they're on their 3rd year of an anti-estrogen drug. That's not really going to change things per se. So you can still do your every 3-month measurement. But when somebody is in the midst of treatment and you're trying, not sometimes, but often you will see changes in the DNA fraction before you're going to see evidence of progression on scan.
Absolutely.
And so you can use it then in the context because they're not going to be off treatment for 4 to 8 weeks in some people. So we can still use that in that context.
Yeah. The MRD world is paradigm changing right now. The amount of data that's being put out in molecular residual disease is just, it's overwhelming sometimes to actually go and read. Now, most of the data, as of right now, is in the CRC space, right? So looking at colorectal cancer. Okay. So looking at MRD in your post-surgical stage 2B resection patients, right? And if you do a small bowel or a bowel resection on a stage 2B CRC patient, and then they come back for follow-up in 4 to 6 weeks and they get an MRD test done, right? The data shows that if that comes back positive, in other words, that particular test shows that there is still tumor-derived DNA present, CRC-specific tumor-derived DNA, the outcomes are extremely poor and there's a near inevitability of recurrence. That's what the data shows in the CRC space. It's similar data in other indications in the lung space, after third line treatments and some ovarian and endometrial. There's data now that's coming up that's showing that same story is happening across multiple indications. Okay. And Jesus. the same with after complete. So let's say someone is stage 2, stage 2B. Let's say they come out of surgery and the clinician orders them on FOLFOX or FOLFIRI or something like that, and they do this chemotherapy, and then after chemotherapy they do an MRD again, and that's still positive. Again, it's 98% chance of recurrence. Yep. Very poor PFS and very poor OS.
Got it. Yeah. So, and that's the MRD, which stands for molecular residual disease, meaning you can't see it on scan, but you can see it on this NGS technique by looking for circulating tumor or cancer tumor DNA in the blood. Yep. And so in the world of, let's just say somebody has cancer, they've been through treatment, they're still scan positive, it shows that there's a little progression. The medical team says, we probably need to change your treatment, but let's do an NGS test. Let's do a liquid biopsy. People are doing that because they're looking for other information that they can entertain that might lead to a therapeutic target for which there's a targeting agent. I mean, that is one of the main precepts of liquid biopsies.
Yeah, absolutely.
What would be a prime example of that? You brought up EGFR, but we have things like ALK, we have things like ROS1, and there are so many different types of molecular inhibitors. We usually call them TKIs or tyrosine kinase inhibitors that can go in and target. And so what are some of the ones that you see most often coming through that are targetable?
Sure. I think probably the best example right now, and it's because this study just published its data, and this is in the breast cancer world. So we'll go back.
So let's date it. When did it publish? 2025?
Yeah, like a month ago.
Okay.
ASCO, right?
Okay.
So yep. And this is the SERENA-6 study. And this was sponsored by AstraZeneca. And this was looking at a new oral SERD, a selective estrogen receptor degrader.
Okay.
And generally, these breast cancer patients, like you mentioned, they're on long-term treatment paradigms, right? They're on an aromatase inhibitor, or they're on some kind of an anti-estrogen therapy or something like that for long periods of time. Yeah. And cancer is a disease of evolution. Right? Cancer is going to change. It's going to evolve as you change the environment. Right? Exactly. Exactly. And so, oncologists are combating evolution at its core. It's a very crazy thing to think about. But you're giving a patient a therapy, and that therapy is then going to change their cancer cells. Right. So this study, SERENA-6,
selection. I mean,
it's right. It's selective pressure. Yeah. Absolutely. You're putting their cancer cells under selective pressure, and inevitably it's going to change. It's going to respond to that pressure. Right. So SERENA-6 was really a landmark study in my mind, because it was one of the first clinical trials that said, hey, we know that ctDNA technology is a thing. Like, there's a lot of data that says we should be using this. So they said, okay, let's look at the emergence, or look for the emergence of a specific mutation in ESR1, right? That these mutations, that's the estrogen receptor. And so if we see an ESR1 mutation, we know that they are now resistant to aromatase inhibitors, right? So we need to change their therapy. Can we detect that before we see progression radiologically?
Can you detect that before somebody's even placed on an aromatase inhibitor?
That's yes. So that's possible, but generally the aromatase inhibitor is what's causing the ESR1 mutation to actually occur. So I guess the key thing here was, let's monitor by ctDNA, right, during the standard of care treatment, and let's see if we can find the ESR1 mutation before we see progression radiologically. Yes, we can. And we've been able to do that. We can do that in CRC, for example, find other mutations before radiologic progression. But if we see an ESR1 mutation pop up via ctDNA, and we change, or we add this new drug,
the SERD,
right? The SERD. If we add this new SERD, does that have a positive influence on outcomes before we see it radiologically? So I think this is the paradigm switch that I think a lot of medical oncologists in the solid tumor space, they're like, when you ask them, what is recurrence?
So people are going to ask, if I have an ESR1 mutation, and that means that I'm resistant to an aromatase inhibitor, does that mean that the estrogen receptor isn't functioning properly? And if so, then why do I have to degrade it? Why do I even need that? Because is estrogen even going to fit like a lock and key into the receptor if it's now mutated.
Yeah. The mutations that are associated with resistance are not loss of function mutations.
Okay.
So they're maintaining the function of the ER pathway. And I think that's an important thing to point out. And that we still need something that can bypass it. Now that it's resistant to AI therapy, we need something else, right? And there was a previous SERD, next generation SERD, that has been approved for ESR1 mutations. But AstraZeneca said... elacestrant, correct. And so AstraZeneca said, camizestrant, can our drug work with standard of care and produce better outcomes if we initiate treatment switching before radiological projection, but as soon as we see an ESR1 mutation? And the answer was overwhelmingly, yes, we can. And if we do this, we can see a 56% lower progression-free survival or 56% lower risk of progression or death.
And so are you suggesting, you're not making medical suggestions from your perspective, but in general, do you see physicians now using NGS to look for ESR1 mutations earlier in people's aromatase inhibitor therapy? Because that's not the norm. And I don't know if medical oncologists are moving towards that or not.
Yeah, I think with this data, for sure, being kind of a nail a little bit in the coffin saying, look, this molecular recurrence thing is real. We should be looking at this and we should be monitoring this. I think they are now.
Okay.
And there's been a lot of excitement in the field when we talk to breast oncologists about this and like, this is great. And now I really see the utility of doing this. And so we are seeing that being a thing.
So sometimes patients will get their NGS report and they'll come to us and they'll say, my doctor said there wasn't anything here that we could treat. And, I look at it, I'm like, okay, I'm a naturopathic oncologist. I'm going to look at this a little bit differently. We see some variants of unknown significance, which people ask me about. What does that mean? Why are they the significance unknown, for instance? And is there something you can do with them? And then we have some botanical inhibitors. I mean, certainly not to the magnitude of any of the pharmaceuticals, and we know that. Sure. We talk to our patients about them. And we can also use polypharmacy, and ours are, mostly devoid of side effects, which means they're not affecting those pathways nearly like they would from a pharmacological model. But variants of unknown significance, I think, are important to talk about. And before we're done, we do need to talk about TP53 since we talked about it earlier. But are there some VUSs or variants of unknown significance that then graduate to become variants of significance?
Yes, there are.
Okay.
And I think, VUSs are an important pool of next generation pathological significant data, right?
Yes.
And I think there's just not enough people that are doing the functional work to say this VUS actually does this to this pathway and it is significant in oncology context, right? But I would say that from my perspective, if you see a patient, let's say they have no actionable oncogenic driver biomarkers, right?
And that means that there's not a medicine that can be used to target them specifically in a clinical trial.
Absolutely. There's nothing that we can target from a therapeutic context, right? But they have a ton of VUSs, right? Yeah. In my mind, I see that and I say that's a lot of genetic instability.
Yes.
There's a lot of mutations happening there in all of these different genes, right? And it, for me, and I'm not a clinician, but I think that would be a great candidate for a platinum-based chemotherapeutic because there's a lot of DNA damage. If we induce more DNA damage, we can maybe do something about that, right? So I think the context of how you see a VUS, I think is important.
Indeed, I appreciate that. And then does that figure into TMB at all?
Tumor mutational burden, yeah, it does. So I think we see that patients that have more mutations and more VUSs reported, I think have a corollary, a correlated, slightly more correlated higher TMB. I don't know if we actually have data that puts statistics on that number, but TMB is just the number of mutations per megabase of DNA.
Okay.
Right. So people who have more mutations that are detected have a higher TMB. People who have lower mutations have a lower TMB.
Yeah, and higher TMB seems to, there's more mutational burden, so there's more stuff going on, and those can be sometimes more sensitive to immunotherapy.
Absolutely.
The immune system, and that doesn't necessarily just mean the conventional, now traditional immunotherapies like the PD-1 or PD-L1 inhibitors, but I think any kind of immune attack, and we certainly work in that field. And as naturopathic doctors, we're going to take a look at all of those parameters and say, okay, this is dirty. What can we do about this? Where can we fit this in your lifestyle? or from some kind of healing parameter? How can we engage a frequency, a nutrient, some type of IV, dietary change, sleep parameter change, neurocognitive issues?
Absolutely.
There's a lot that, and none of this is data driven, but this is certainly just makes sense from a biological, perspective.
You're optimizing the environment.
Yeah,
I mean,
we have to, and we're
environment.
trying to also reduce further mutations too, because they can get more dirty, and once they're dirty, they kind of get more dirty.
It's just a cycle, right?
Do you like what you do?
I do. I love what I do.
And do you like, as you look into the future of what you do, is it exciting?
It is. Yeah. It's a very exciting time. There's so much data coming out, and I think I love to talk about science, and I love to talk about science with anybody about anything. And I think that the data coming out right now in precision medicine, specifically in oncology, with AI-driven drug discovery and all these things, there's just the number of FDA approvals for targeted therapies is just skyrocketing. So I think it's a very exciting time. Now, it's going to be a very confusing time for medical oncologists. There's going to be 15, 20 different drugs to treat all these different mutations soon. And it's going to be, I think, the role that people like me are going to play in medical affairs to help physicians and clinicians figure all this stuff out, I think is only going to get more important.
Do you think you're going to see optimized regimen suggestions on the reporting formats?
I do. And I think that's going to have to be data driven. So I think as we start seeing more RCTs and things come out, I think that will come about.
Yeah.
Yeah.
You talk about RCTs or randomized clinical trials, which is taking groups of people, but traditionally they've been large groups of people, so you can get a statistically significant N. But we're actually right now talking about N of 1. As we see these VUSs and these complex manifestations of one's own single tumor burden, I'm wondering if the sample sizes for these RCTs, or maybe we'll see somewhat of a departure from an RCT and we'll go into the world of human biology, knowing that we just can't we already are randomized, each one of us. And as an ND, I really respect and look at human individual bio and physiological individuality. And that's what we're seeing, is that a cancer is not really a single cell type, but it's a complex manifestation inside of one's own body, the development of which we're not necessarily sure of, but we can see these on these NGS tests, that how it's presenting. And you may even see the advent of what was once failed polypharmacy in the conventional world being brought into maybe we can optimize these regimens. Maybe you don't, if we want to use, 2 TKIs at once, maybe we use them sequentially or at different parts of the cell cycle or different parts of the treatment cycle. We don't, to reduce toxicity and enhance cell kill. So I'm wondering if you think that that's coming as well.
I think that there's a lot of excitement in that realm because, and I'm also a bioinformatics person. I love computational biology, and I think bringing AI into this fold, into the fold here, will say, hey, maybe we'll be able to tease apart the complex biological interactions that these VUSs have a play in, right? And maybe we will start to be able to tease apart this from a biological foundational level to be able to do a lot of the things that you're suggesting. Because you're absolutely right, everybody's cancer is different, and everybody's cancer is heterogeneous in terms of its genetic profile and the other alleles and variants that are there. So I think it's a complicated situation, which I'm hoping that AI will help us deconvolute.
Yes, and it's interesting because cancer is evolving and changing, but at the end of the day, cancer is seeking to destroy itself by killing the host, right? If you let it go. But I call it a big dumb warrior. It's big. It's a warrior because it's always trying to battle through the treatments and break through, but it's dumb at the end of the day because it could also just live in the body too and not cause some problems. And
see, I think about it in the opposite way. I think cancer is smart.
Okay.
I think it's incredibly smart.
Because it's able to mutate.
It thinks so much faster than a normal human cell because of the innate changes in its biology. And that's the hard part, is how do we outsmart it? How can we be smarter than a person's cancer? And unfortunately, most of the time we can't.
And I don't know that we're going to have that answer in the world of pharmacognosy. I don't know that that's where the intellect is going to lie.
I agree.
Yes, and I say scientist to naturopathic clinician, I want to work with you on this. I like it.
Yeah.
So does this mean that as we move towards that era of utilizing these VUSs and really coming to conclusions about how to treat people and maybe some of these evolved regimens, that more clinicians just need to run more of these tests on people?
I don't understand from a scientific perspective how any clinician can make an informed decision about their therapeutic choices these days in oncology without running an NGS.
Done.
Bottom line.
Done.
Yeah.
Wow. Fascinating.
Yeah.
I can't wait till part 2 of this sometime when we get to sit down again and have our next discussions.
Absolutely.
But folks, it's been my pleasure to sit with Dr. Brandon Guida to talk about all of these different terminologies and things that we talk about that you probably experience yourself or a loved one has if you've held their test result. There's so much information in there. So I appreciate you coming and spending some time with us.
So much fun.
Yeah, this has been great.
Absolutely.
So for Rubin On Point, be well.
Thank you for joining us on Rubin On Point, a podcast about cancer, resilience, and most of all, about being human. I hope today's conversation inspired you to see new possibilities for growth, healing, and strength in your life. If you enjoyed this episode, be sure to subscribe, leave a review, and share it with someone who could use a little extra resilience in their day. Remember, no matter what challenges you're facing, resilience isn't just about bouncing back. It's about building a life of purpose, connection, and vitality. Until next time, stay strong, stay healthy, and take care of yourself and those around you.